Hello friends, welcome to CEC Live Lectures. Dear friends, with our ongoing series on cell biology, today we are going to talk on plasma membrane. And for the discussion on the topic, we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is an eminent professor of zoology, and she is currently teaching as assistant professor in Department of Zoology, Ramjas College, University of Delhi. Dear friends, Dr. Charu Dogra Rawat has immense experience, and her skills always help students in understanding various topics in detail, and we believe that today also we are going to get in-depth knowledge through her. So let's welcome our guest, Dr. Charu Dogra Rawat. Hello ma'am, welcome to the lecture.
Thank you. So when we talk about cell basically means a containment. Cell refers to a compartment. So when we talk about the containment, or when we talk about a compartment, then it needs to have a certain boundary. As simple as that. So that boundary, what we know in the terms of scientific knowledge or in terms of cell biology, is the plasma membrane. Now this plasma membrane is not a dead wall which contains all the contents which are inside the cell, but it's a kind of a physiological barrier. It does allow many movements across this plasma membrane, which is very important for the sanctity of the cell. So today's session is dedicated to understand basically the structure of the plasma membrane, in which we will particularly talk about the studies, the brief history of the studies on the plasma membrane structure, that how the present understanding of the cell membrane structure, how it was deduced, and we'll also talk about the chemical composition of the plasma membrane. The two references which is used for this particular session are the book called as by Karp, which is Cell and Molecular Biology Concepts and Experiments, 7th edition, and Thermal Biophysics of Membranes by Thomas Hamburg. So these are the two references from where the material has been taken. So the cells are separated from the external world by this thin, fragile structure called as the plasma membrane that is only 5 to 10 nanometers wide. So it's a very thin membrane. No hint of the plasma membrane is detected, which is quite obvious when a section of a cell is examined under a light microscope. So light microscope's magnification and resolution does not allow us to view the cell membrane under it. In the second half of the 19th century, it became evident that an osmotic barrier separates the inside and the outside of the cell. So how did they come to know about this physiological barrier? We remember that when the cell was discovered, Robert Hooke just identified basically the compartments in a cork, and he termed it as cells. So these compartments and these things are actually enclosed by a membrane, came to be known, in the second half of the 19th century, when actually it was observed that the cell allows certain molecules to be passed through it, however does not allow certain other molecules. So the plant cell protoplasts or the cell cytoplasm were permeable to water, but to larger macromolecules, not to the larger macromolecules like sucrose. So it's a selectively permeable thing which is surrounding this cell protoplast. So what is it? So however, nothing could be observed under a lighter microscope because the resolution was too less, and later on we got to know that this particular membrane which is enclosing the cell is very, very thin. So at that point of time they knew that there is some barrier which is allowing this selective permeability across the plasma membrane, across the cell, but they didn't know what it is. So Pfeffer was the scientist who was the first to study the osmotic pressure within the cells and formulated the idea that actually this osmotic selective permeability is because the protoplasm of the cells is surrounded by this thin layer which is called as the plasma membrane. This membrane does not only cover the outer surface of the cells, but it also separates all aqueous environments of different composition from each other. So not only the outer boundary of the cell is enclosed by this particular thin membrane, but inside the cell, as we got to know that there are different aqueous environments, which we call them as cell organelles. So there is mitochondria, there is nucleus, in case of eukaryotic cells, or there are different, the chloroplast and plant cells. So all these aqueous environments are basically enclosed by this particular thin membrane, and it was identified, or it was basically observed by the experiments done by the scientist Pfeffer. We'll just quickly go through a brief history of studies on the plasma membrane structure. It dates back to 1895, when Robertson, the scientist, investigated the osmotic properties of cells and noticed that the permeation of molecules through membranes is related to the partition coefficient between water and oil. So studying this osmotic selective permeability, that how certain molecules can go across and what is the partition coefficient for this mechanism to occur, he found that this coefficient was actually related to when things pass across the water and the oil layers. He was the one who led, this observation led him to hypothesize that the thin membranes surrounding the cells have the properties of oil. So that was the first observation, that there are certain oil molecules which are basically composing what is called as the plasma membrane. He called the layer surrounding the cells the lipids, because it was made from the oils or the lipids and the cholesterol. So that observation led to the first reference of the outer boundary of the cell membrane of the cell to be lipoids, because they compose mainly the lipids. Then came Langmuir in 1917 and Gottel and Grendel in 1925. Langmuir actually developed an apparatus in which molecular layers of lipids were separated at the air-water interface. So it was something like this. So it had got a surface where the oil or whatever biofilm can be poured, and then that film, there is an interface between the water and the air, or the surface of the layer and the air, and you can see that there are two clamps or two barriers which can adjust the size of this particular layer. So he developed this apparatus in which the molecular layers of lipids were separated at the air-water interface. With this monolayer, through the lateral pressure, the lateral pressure of the monolayer films could be measured. So what he could do was he could measure the lateral pressure of the monolayer. So as soon as the oils were first extracted and then poured onto this trough, and when it is poured onto this trough, they form a monolayer or a single layer on the surface of this particular instrument. So on that layer, the lateral pressure could be measured by adjusting the bars which are there. So when we move them across, then it will take the least distance which is in between the film, and therefore this pressure can be measured. Langmuir proposed that in the molecular film, the polar head groups were directed towards the water, whereas the hydrophobic hydrocarbons are pointed towards the air phase. So he not only could calculate the pressure, the lateral pressure of the monolayer films, but he could also identify that this particular layer has actually two components. One is the head groups which were directed towards the water surface, and the other hydrophobic groups of the oil were basically towards the air phase which was there. Two Dutch scientists, Gotter and Grendel, they extracted the lipid from the human red blood cells and measured the amount of surface area the lipid would cover when spread over the surface of the water. So they made use of this Langmuir film balance, and they extracted the lipid layer or the lipids from the human red blood cells and measured the amount of surface area this lipid would cover. They found that the surface area of the monofilm was within error exactly 2 times that of the cells. They concluded that cell membranes are actually made of 2 opposing thin molecular layers, because it was basically 2 times whatever that is extracted from a monofilm layer or whatever it is found, so based into the error. So it ranges from 1.8:1 the ratio, or 2.2:1. So they concluded that it is actually a bilayer. The ratio of the lipids is actually 2:1. They are made of 2 opposing thin molecular layers. This double layer is constructed such that, as stated by Langmuir or as observed by Gottel and Grendel, which is basically that the hydrophobic ends were towards the air phase and the hydrophilic polar heads were towards the water phase. So based upon that, they said that this double layer is constructed such that two lipid layers form a bilayer with the polar head groups pointing towards the aqueous environment. So in this structure you can see that these are the individual molecules. This is the polar head and this is the hydrophobic chain. So they are arranged in the lipids are arranged in such a manner that there is a bilayer, because the ratio was coming twice. These polar head groups are actually towards the aqueous environment. You can see the water here, and these hydrophobic chains are towards the inside. So according to these two Dutch scientists, the lipid bilayer or the lipid double layer was supposed to be looking like this. And this is again, they use this instrument and they calculated the surface area of the lipid. So when the lipids are extracted and poured onto this Lamguer trough, then they form such a kind of a monolayer. So you can see there is an air and there is water. So this is the waterfront where the polar heads have oriented them towards, and this is the air where the hydrophobic chains have oriented them towards. And when they calculate the ratio, they found that in the cell there is actually a bilayer. So it's kind of this kind of a structure. This is a typical structure of a lipid molecule where there is a polar head and there is a large hydrophobic chain. This is actually a simulation of a fully hydrated lipid bilayer. The phospholipid head groups, which are orange in color, the water molecules are blue, and the white fatty acid chains are green. So you can see that this is representing actually a bilayer. This is a waterfront, therefore the polar heads, which are orange in color, are directed towards this waterfront, and the hydrophobic chains, which are green in color, is actually towards inside. So on both the fronts, the hydrophobic chains are towards inside and hydrophilic polar head groups are towards the outside. So they could make out by this using this Langmuir film balance that such a kind of an arrangement of the bilipid layer is there. Then came the Danelli and Davson in 1935 with what is called as their sandwich model. The lipid solubility was not the sole determining factor as to whether or not a substance could penetrate the plasma membrane. So that was their observation, that there has to be something more than lipid molecule. Lipids cannot be there to to reason the transport of all the molecules across the cell membrane. So there has to be something more. Another point was that the surface tensions of membranes were calculated to be much lower than those of the pure lipid structures. These 2 points clearly indicated that there is something more than lipid in the bilayer membrane which is there. So Jim Danelli and Hugh Davson proposed a model of the cell membrane consisting of a lipid bilayer, and on both the sides they have a very tight protein layer associated with it. So you can see this is the lipid bilayer, and on both the sides there are protein layers which are present. So you can see that these protein layers again have 2 components, where the water permeable part of the protein is towards the outside and hydrophobic part of the protein is towards the core or towards the inside, and this is a typical lipid molecule. So such a kind of arrangement is shown, that there is a lipid hydrophobic bilayer inside, and then on both the sides there is a tightly associated protein layer which is there. They used their model to interpret the observation of different membrane permeability of ions and hydrocarbons, which could not be explained by keeping the plasma membrane as only the bilipid layer. So they tried to, because they were concentrating on describing the cell permeability to various ions and biomolecules. So based upon that, their structure, the sandwich model which they proposed, they tried to explain the observations of the different permeabilities for ions and hydrocarbons, and for that they make certain consideration, that they assume that the membranes are both lipophilic and hydrophilic character. So it's not only oil loving, but they are also water loving character the membrane has. The water containing regions in the membrane give rise to the ion transport depending upon the water content of the membrane and the charge on the ions, and the lipophilic parts are responsible for the transport of water insoluble molecules. So purely lipid molecule would have been only the hydrophobic or water insoluble molecule loving, but they propose that actually it contains both the hydrophilic as well as the lipophilic part, and therefore it allows the transport of various kinds of ions and molecules across it. So Davson and Danelli also suggested that in addition to the outer and inner protein layers, the lipid bilayer was also penetrated by protein lined pores. So you can see here that these are the molecules which are the lipid molecules, and outside they have protein associated with it. So lipophilic as well as the hydrophilic molecules can be passed through. They can also pass through, the hydrophilic molecules can also pass through such pores that occur in the biological membrane, which are lined by the hydrophilic proteins. So the lipid bilayer was also penetrated by the protein lined pores, which could provide the conduits for polar solutes and ions to enter and exit the cell. Because the polar molecules cannot pass through the hydrophobic lipid membrane, therefore there are certain protein pores which causes these hydrophilic molecules or polar molecules also to pass across the membrane. So this was the Danieli-Davidson sandwich model, where there is a lipid bilayer and then outside there are protein layer on both the sides. So they are sandwiched, the lipid bilayer is actually sandwiched between the protein layers which were there. Then came the Robertson model. As we said that, till now the plasma membrane, they were all indirect evidences, because the plasma membrane could not be observed under the microscope. The light microscope magnification and resolution is so less that it could not identify or it could not make the observation of the plasma membrane. But now till like 1950s, the advancement in the electron microscopy was coming, and Robertson actually collected the electron microscopic images of many cells and organelle and gave rise to what is called as the unit model. So you can see here that these are the two opposing plasma membranes showing the double layer character. So this is one membrane, this is another membrane, and this is actually the intercellular space. So this one membrane contains a trilaminar structure, where you can see that these are the two thin lines, which is basically the lipid bilayer which they are talking about. So it's a double layer character that they are showing, supported the view of the Gortel and Grendel and of Danieli and Davidson. So whatever they have proposed, they actually kind of gave a very foolproof evidence through these electron microscopy, where they observed these plasma membrane structure from various cells and various organelles. Robertson's conclusion was that all evidence points at a common construction principle for all membranes of biological cells. So they talked about that the normal construction principle, and their model was actually very much related to the Danieli and Davidson model, but it had now a very authentic proof behind it, and they said that there is a common characteristic of all plasma membrane. Although their model was misinterpreted to consider that all cell membranes perform the same kind of function, but they never talked about any function. They only talked about that there is a common structural plan of the plasma membrane for all the cells and organelles, which basically is that they form a three-layered structure, are about 7.5 nanometers thick. In Robertson view, the protein layers are adsorbed to the lipid bilayer. So you can see again here, this is a lipid bilayer, and these are the two protein layers which are adsorbed on both the sides. So this model actually is very much similar to the Danieli and Davidson model, but as I said, that one thing it pointed out was that it was backed up by a very authentic proof. And second point was that they said that this is the common structural plan of all the membranes. Obviously there could be variations, there could be functionally related variations, and they never said that they were functionally, every membrane has this kind of a structure, but they said that all the cell membranes have this kind of a structure which is there. In fact, they also, they form this three-layered structure or trilaminar structure consisting of a lipid bilayer and the protein layers outside and inside, both the facing thing there is there. Then in 1972 came the fluid mosaic model of Singal and Nicholson. The model can be summarized as follows. The membranes are constructed from lipids and proteins. So the two important components of plasma membrane is the lipids and the proteins. The proteins form mainly two classes. One is the peripheral protein, which are those proteins that are only loosely attached to the membrane surface and can be easily separated from the membrane by mild treatment, for example, cytochrome C in mitochondria or spectrin in erythrocytes. And the second is the integral protein. So now what they talked about is that there is a lipid bilayer, no doubt, but apart from, in spite of having this adsorbed protein layer on both the surfaces, the proteins are actually of two different types. The peripheral protein, which are actually on the outside of this lipid bilayer, but they are loosely attached and they can be easily removed. However, there are another set of proteins which are actually embedded inside the plasma membrane. So the integral proteins, in contrast, cannot easily be separated from the lipids, and they form the major fraction of the membrane proteins. So the structure forming unit or the matrix is the lipid bilayer or the lipid double layer. The proteins may be either adsorbed to the membrane surface or span through the membrane. So this was the model which is, currently the common structure of the plasma membrane relies on this particular model, and it is true till now that there is a lipid bilayer and there are these two different kind of proteins which are there. One is the peripheral protein loosely lying outside, and there are other integral membrane proteins which spans this lipid bilayer also. So this is the kind of structure you can see. So this is a fluid mosaic model. You can see this is the lipid bilayer. So lipid bilayer is basically the matrix. This lipid bilayer is the matrix, and these are the two different kind of proteins which are found. So some proteins, as you can see here as well as here, are the transmembrane proteins. They pass through the entire length of the membrane. So they are starting here and ending. So they are the transmembrane proteins. And then these are the globular proteins which are, protruding from one side, and they might be loosely attached. So there can be some here also. So these are some can be the peripheral proteins which are just on one side. There can be other proteins which are not at all embedded inside the lipid bilayer but just lie on this lipid bilayer, and then there are certain proteins which are embedded and which will be passing or spanning through the entire plasma membrane. So the lipid bilayer, including this globular protein, intercalated with the membrane and transmembrane proteins. The membrane proteins are not at all distributed homogeneously. So till now we saw that the lipid bilayer is there and the proteins were adsorbed on both the sides in a very homogeneous manner. So fluid mosaic model tells that it's not like that. Mosaic actually means, in fact, and Danieli and Davison has also speculated that their proteins might be arranged in a very mosaic manner, and they did mention that, but they didn't bother about that because they were trying to talk about the reasonings for the cell permeation, and therefore they came up that apart from lipids, the proteins are also associated, and very plainly and simply they said that proteins must be present on both the sides of the bilayer membrane. But they did mention a little bit about the mosaic model, but it was actually established by this fluid mosaic model by Sanger and Nicholson. So mosaic basically means that the membrane proteins are not distributed homogeneously, but they are distributed very inhomogeneously and therefore have a mosaic kind of appearance. Fluidity basically means that the lipid membranes of the biological cells are in the fluid lipid state. With examples, for example, the myelin sheath, where they are not fluid, but apart from that, most of the biological membranes contain the lipid part as the fluid in nature. In fact, the protein part is also fluid, in which the proteins can freely diffuse. So it's not a very static structure, it's very dynamic structure. And that dynamicity is provided by that the lipid molecules can have various kinds of movements. And therefore it is called as a fluid model, because the lipids are, they can, even the proteins can move within the lipid molecules, and in fact the lipids can also move from one side to the other. They are also not very stable. So the plasma membrane is very, very dynamic. So this was the fluid mosaic model, and the fluidity was basically, the evidence for this fluidity or support for this fluidity came by this Fry and Eden's experiment in 1970, on which the fluidity was based, because they showed that when two different cells with two different proteins are forced to fuse, the proteins redistribute over the whole surface within 40 minutes. They took a mouse cell, they took a human cell, they labeled the mouse cell with one fluorescence, say red fluorescence, and they labeled the human cell with another fluorescence, a green fluorescence, and when they used Zendivirus, for example, for causing the fusion of these two cells, the two cells got fused, and within an hour, even less than 40 minutes, they see that the fluorescence was actually redistributed on the fused cell. So it was kind of mixed, and that could only happen because the proteins were fluid in this lipid plasma membrane. So this plasma membrane had this fluidity in their thing. So Singer and Nicholson also noted that the fluid mosaic membrane is most likely asymmetric and displays distinctly different features on the inside and the outside of cell. Asymmetry of membrane lipids has in fact been found in experiments such as Rothman and Leonard and Rothman and Kennedy. They did experiments which talked about the asymmetry of the membrane lipids. And also proteins are now known to display preferential orientation in the membranes. We'll talk in detail about this asymmetry when we talk about the function. So asymmetric, the lipids are also asymmetric as well as the proteins are also asymmetric. Just quickly, the last kind of model is basically the Mattress Model by Robertson and Bloom, which suggests that proteins and lipids display interactions with a positive free energy content due to variations in the hydrophobic length of the molecules, and this interaction between protein-protein molecules of a plasma membrane or protein-lipid molecule of the plasma membrane, or in fact lipid-lipid molecule of the plasma membrane, results in the formation of the domains and the clusters. So plasma membrane is not just, the structure is not like that it is fluid, but it is not in relation to anything with each other. So refinement of the fluid mosaic model of Singer-Nicholson, which takes into account that lipids or proteins may distribute inhomogeneously and then domains and clusters may form within the membrane. So here you can see that some proteins can be larger than what is the width of the plasma membrane. So either it has to protrude out or they have to do some kind of adjustments there. So they interact and then form such kind of domains and clusters which are there. Such kind of interaction is termed as hydrophobic matching. If the hydrophobic core of a membrane protein is longer or shorter than this length, either some hydrophobic proteins or lipid segments are exposed to water, or the lipid membrane has to be deformed to compensate for the unfavorable hydrophobic interactions. Due to this hydrophobic matching, various domains and clusters will appear. So that was a brief history of the structural models of the plasma membrane. As we know now that it contains a lipid bilayer, various kinds of proteins which are asymmetric on both the sides, and there is an interaction between the components of the protein giving rise to the various domains and clusters in the plasma membrane. Thank you.
With this note, thank you, ma'am. Thank you so very much for giving us this productive session. And friends, there is a lot more for you. So you are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Dear friends, as you know that today we are talking on plasma membrane. In our previous session we discussed on the structure of plasma membrane, whereas in this half we are going to discuss on the composition of plasma membrane. And for the discussion on the topic we have with us in our studios Dr. Charu Dogra-Rawat. Dr. Charu Dogra-Rawat is a professor of zoology, and we believe that you are getting maximum knowledge through her. So let's welcome our guest once again, and let's request her to give us more and more details about the plasma membrane. Hello ma'am, welcome to the lecture.
So biological plasma membranes, as we went through the brief history of the discovery of the structure of the plasma membrane or deduction of the structure of the plasma membrane, we saw that the plasma membrane is composed of a common structural unit, a matrix, which is the lipid bilayer. Then these lipid bilayer also has certain proteins, and these proteins are of two types, the peripheral proteins and the integral proteins. So peripheral proteins are loosely bound, and the integral proteins actually inserts into the lipid bilayer and sometimes can also span this lipid bilayer and called as the transmembrane proteins. So now we will quickly go through the different kinds of lipids which are found in the plasma membrane, different types of proteins which are there, and other molecules such as the association of the carbohydrates with the lipids and the proteins. Basically, we will talk about the composition of the plasma membrane. So the biological, quickly going through this picture, which is actually telling about the structure which we come across and which we know now that the plasma membrane has. The biological membranes contains hundreds of different lipid species with variable head group and chain composition. Most lipids possess two apolar hydrocarbon chains with variable length. Furthermore, lipid membranes undergo melting transitions which are accompanied by changes in the effective lipid chain length. Thus, also the lipids within the membrane plane can form various clusters, domains and aggregates. And for now, this we have agreed upon, that a modern view of the biological membrane has to include the phase behavior of the lipid and protein components. So we see that this is the bilayer. It has got an apolar molecule on the top and a very polar molecule on the top and an apolar hydrocarbon chain, and then there are proteins which are there. And these proteins, based upon the difference in the phases, should come in the clusters, domains and aggregates form also. So this is the typical structure of a biological membrane which we now understand. Looking at the chemical composition, as we said, biological membranes display complex compositions with hundreds of different lipids and proteins. The ratio of lipid to protein always varies depending upon the type of the cellular membrane, that whether it is the plasma membrane or the membrane of an organelle such as endoplasmic reticulum or membrane of Golgi, the type of organism, that whether the bacterial plasma membrane we are looking at or plant or animal, and the type of the cell, for example, it's a cartilage cell, muscle cell, neural cell, what kind of a cell it is. So we talked about that the lipid is bilayer, where the ratio is 2:1 between the lipid and the entire of the cell. But the ratio of lipid to protein actually varies. So they are not in the equal composition, they are not in 2:1, but they varies according to the cell type, according to the organism from where the cell is coming, and as well as according to that within a cell what the membrane is of what part, whether it is the plasma membrane, the outer plasma membrane, or whether it is of any other organelle. And I must tell you that this ratio is basically functionally dependent. So when we will talk about the functions, you will see that that certain cells will have more of proteins because they need such kind of functions, because they will have a lot of receptor molecules which are present on the cell, and the receptor molecules are all proteins. Or you will see that certain molecules will have a lot of lipid molecules which is there, and the protein ratio will be less. So we will talk about all these different and will correlate these differences in the ratio with the functions of the plasma membrane when we go into the functions part. To a large degree, these differences can be correlated with the basic function of these membranes. Membranes also contain, apart from these two biomolecules, the lipids and proteins, the carbohydrates, which are attached to either of them. They can be attached to the lipids or they can be attached to the proteins. So we will talk about in detail about what types of lipids, what types of carbohydrates and what type of proteins we are looking at. Starting with the membrane lipids. Membrane contains a wide variety of lipids, all of which are amphipathic in nature. Amphipathic, which basically means that they contain a hydrophilic part as well as a hydrophobic part. Phosphoglycerides, most membrane lipids contain a phosphate group, which makes them phospholipids. Because most membrane phospholipids are built on a glycerol backbone, they are termed as phosphoglycerides. So, most of the lipids of the plasma membrane are the phosphoglycerides which are there. Membrane glycerides are diglycerides, where only two of the hydroxyl group of the glycerol are esterified to a fatty acid. Triglycerides, all the three alcohols are esterified to the fatty acids, but in plasma membrane they are diglycerides, where only two of the hydroxyl groups are esterified to the fatty acid, and the third is esterified to a hydrophilic phosphate group, and therefore these phosphoglycerides has a polar head and there is a large hydrocarbon or fatty acid chain which is there. So, two of the alcohols has this large hydrocarbon chain and one has this phosphate group which constitutes the polar head. Membrane phosphoglycerides has an additional group linked to this phosphate. So, this polar head might contain additional groups, most commonly either the choline, then it is termed as the phosphatidyl or PC, ethanolamine, forming what is called as the phosphatidyl ethanolamine, referred to as PE, serine forming as phosphatidyl serine, or inositol forming the phosphatidyl inositol. Each of these groups are small and hydrophilic, and together with the negatively charged phosphate to which it is attached forms a highly water soluble domain at the one end of the molecule, which is called as the head group. So, you can see the structure here. So, if this is the molecule, the lipid molecule, then this is the head part which we are talking about, and this is basically the hydrophobic part or the tail end which we are talking about. So, this hydrophilic part which always faces the waterfront can constitute any of these. A normal one, a glycerol thing which is esterified to various acids, they are termed as the phosphatidic acid. But in this case, you have these phosphate groups, and these are the hydrophilic chains, hydrophobic chains which are there. So, R basically R refers to the various hydrocarbon chains which are associated with it. So, they can be long chains that are associated with it, and this phosphate group can be associated with different molecules. Phosphatidyl choline, this is choline, phosphatidyl serine, phosphatidyl ethanolamine, and phosphatidyl inositol. So, these phosphate groups can be attached to these additional molecules. This phosphate group, negatively charged phosphate group, as well as these molecules actually form what is called as the head of the polar head of the lipid. And this chain which we are talking about, this hydrocarbon chain and this glycerol component is basically the hydrophilic part which is there. So, you can have a look at this, that this is how the lipids, various kinds of lipids of a plasma membrane look like. A hydrophilic part which is facing towards the outside, they can be negatively charged or they can be neutral, and a hydrophobic part which is basically towards the interior, having these long hydrocarbon chains which are there. At physiological pH, the head groups of phosphatidyl serine and phosphatidyl inositol have an overall negative charge, whereas those of phosphatidyl choline and phosphatidyl ethanolamine are neutral. In contrast, the fatty acyl chains are hydrophobic. Unbranched hydrocarbons approximately 16 to 22 carbons in length. A membrane fatty acid can then be saturated, lacking a double bond, monosaturated, containing only a single double bond, or a polyunsaturated molecule containing many double bonds which are there. Phosphoglycerides present in the plasma membrane often contain one unsaturated and one saturated fatty acyl chain. So, this R group can be 16 to 22 nucleotides, 16 to 22 hydrocarbon chain length, and these 16 to 22 usually one of the chain is saturated and another chain is unsaturated which contains the double bonds which is there. With fatty acid chains at one end of the molecule and a polar head group at the other end, all of the phosphoglycerides exhibit a distinct amphipathic character. So we talked about this amphipathic character having both the properties of a hydrophilic as well as a hydrophobic molecule. Another class of phospholipids, another class of lipids which are found in the plasma membrane are termed as sphingolipids. They are less abundant class of membrane lipids and they are derivatives of sphingosine, an amino alcohol that contains a long hydrocarbon chain. So you can see the structure of the sphingosine here. So this sphingosine is basically an amino alcohol. This is an amino acid and alcoholic in nature, and a long hydrocarbon chain which is attached to it. Sphingolipids consist of the sphingosine. So this is the sphingosine moiety linked to a fatty acid which is the R component by its amino group. And this molecule, so you can see by its amino group, this is attached to this R molecule or a long chain fatty acid, and such a molecule is termed as the ceramide. So this is the basic unit of sphingolipid in a plasma membrane which is there, which is called as the ceramide. Various sphingosine based lipids have additional groups esterified to the terminal alcohol group. Now this alcohol group will contain many other additions. If this substitution is phosphorylcholine, then the molecule is called as sphingomyelin. So if it is basically phosphorylcholine, then it is sphingomyelin, which is the only phospholipid of the membrane that is not built with a glycerol backbone. Substitution is a carbohydrate if the substitution. So this is containing a phosphorylcholine. It is also containing a phosphoryl group. And as we saw, all the phospholipids were made on the glycerol backbone. However, sphingomyelin is the only lipid which is not made on the glycerol backbone, but instead made on the ceramide or the sphingosine as the base molecule. Now, if this substitution is a carbohydrate, the molecule is termed as a glycolipid. If the carbohydrate is a simple sugar, the glycolipid is called as a cerebroside. So this is basically a ceramide region, which is this, and containing a simple carbohydrate such as galactose. So it is termed as a cerebroside. If it is a small cluster of sugars that include sialic acid, then it is called as a ganglioside. So these are the different terminologies, and these are the different kind of sphingolipids. Also, sphingolipids is a smaller class of lipids, but they are present. The base molecule is the sphingosine. Sphingosine, when is attached to the long chain fatty acid through its amino terminal, is termed as a ceramide. This ceramide can have additional attachments. If this attachment is a phosphoryl group, phosphorylcholine for example, it's sphingomyelin, and if they are attached to different carbohydrate moieties, then they are termed as a cerebroside or the ganglioside which is there. So this is the second class of membrane lipids which is present. Since all sphingolipids have two long hydrophobic hydrocarbon chains at one end and a hydrophilic region at the other end, they are also amphipathic in nature and basically similar in overall structure to the phosphoglycerides. The fatty acyl chains of sphingolipids, however, tend to be longer and more highly saturated than those of phosphoglycerides. So that is the difference between sphingo and obviously the composition of sphingolipids and phosphoglycerides. But as far as the plasma membrane is concerned, the property of amphipathy is retained, and the sphingolipids also have a hydrophobic region and a hydrophilic region which is there. The nervous system is particularly rich in the glycolipids. The myelin sheath contains a high content of a particular glycolipid which is called as the galactocerebroside, which is formed when a galactose is attached to the ceramide. Mice lacking the enzyme that carries out this reaction exhibit severe muscular tremors and eventual paralysis. So it forms a very, very important component of the nervous system which is there. The third molecule, the membrane lipid, which is present in the plasma membrane, is termed as the cholesterol. So another lipid component of certain membrane is the chiral cholesterol, which in certain animal cells may constitute up to 50% of the lipid molecules in the plasma membrane. So you can see the cholesterol molecules are oriented with their small hydrophilic hydroxyl group towards the membrane surface and the remainder of the molecule embedded in the lipid bilayer. The hydrophobic rings of a cholesterol molecule are flat and rigid. So this is a bilayer, this is the example, this is the picture which is showing this is a lipid bilayer, and these green portions are basically the cholesterol. So and these are the hydrophobic rings, and hydrophilic part will be here towards the outside. So this hydrophobic part of the cholesterol, these rings are very flat and rigid, and they interfere with the movement of the fatty acid tails of the phospholipids. So we have already talked about that the lipids also undergo motion. So transition, transversion, various kind of motions, but that retains the fluidity of the membrane. So these portions are becomes rigid. Why bad cholesterol is bad? Because these cholesterol interferes with the fluidity of the lipids, lipid chains, which is basically there. The placement of cholesterol molecules interferes with the flexibility of the lipid hydrocarbon chains, which tends to stiffen the bilayer while maintaining its overall fluidity. Unlike other lipids of the membrane, cholesterol is often rather evenly distributed between the two layers. So you can see here this green region is basically evenly distributed. So if the deposition of cholesterol is much more in the plasma membrane, then the plasma membrane tends to become more and more rigid, and the flexibility is loosened. And therefore, we talk about when the deposition of cholesterol is there, then the dispensity of the membranes is reduced. The fluidity is reduced, and they become much more stiffer, and therefore they cannot widen, and they keep on becoming narrow and narrow. If it is, for example, the plasma membrane of a vessel, so they keep on becoming narrow and narrow because it cannot give that distensibility which can be attained because of the fluidity of the plasma membrane. So cholesterol molecules, they are oriented in such a manner. This is the lipid composition of some of the biological membranes here. So you can see in human erythrocyte, the phosphatidic acid is 1.5. In phosphatidyl choline is more, ethanolamine is this much, phosphatidyl glycerol is absent. However, it is present in the bacterium plasma membrane. Phosphatidyl serine is absent in E. coli, phosphatidic acid, choline is absent. Major part is formed by the phosphatidyl ethanolamine in the E. coli, and this is basically present in the cell organelle, which is mitochondria. So there phosphatidyl choline is in high content, followed by phosphatidyl ethanolamine, and then cardiolipin is also very high. However, serine is less, rest of them are not there, and in fact the cholesterol is there. So cholesterol is there in the human erythrocyte, and human myelin also present. In human myelin, the phosphatidyl glycerol is not there, and you can see the composition of the human myelin. Serine is constant, sphingomyelin is lesser in myelin sheath, however much more in the human erythrocyte, which is there and absent in mitochondrial and E. coli plasma membrane. So this is basically the values are given in the weight percent of the total lipid. And therefore, this is a lipid composition. So as I told earlier that the composition is different in different plasma membranes. So you can see in erythrocyte and myelin from the same organism, or in mitochondria, or a different organism altogether, which is E. coli, the composition of the lipid varies. The ratio of various types of lipids is also varying, and this variation can be associated with the functions of the plasma membrane, or it depends upon the kind of organism and what role is it playing, and what kind of things where they are present, and what kind of functions they need to perform more. This lipid composition can vary in different organisms. Coming to the second biomolecule, which is the membrane carbohydrates. Depending on the species and the cell type, the carbohydrate content of the plasma membrane ranges between 2 and 10% by weight. More than 90% of the membrane's carbohydrate is covalently linked to the proteins to form what are called as glycoproteins. The remaining carbohydrate is covalently linked to lipids to form glycolipids. So both the kind of molecules occur in a plasma membrane. However, higher number is of the glycoproteins, where these small carbohydrate moieties are attached to the proteins of the plasma membrane, but they can be attached to the lipids also, forming what are called as the glycolipids. The addition of carbohydrate to protein is one of the modification of the protein, and it is termed as the glycosylation. All of the carbohydrate of the plasma membrane faces outward into the extracellular space. or into the internal cellular membrane, always towards the cytosol. So they are never inside the plasma. The oligosaccharides, they are always protruding outside into the external environment or towards the cytosol, that is inner bilayer membrane. It is pointing towards the inside of the membrane. The carbohydrate of glycoproteins is present as short, branched, hydrophilic oligosaccharides, typically having fewer than about 15 sugars per chain. Though the oligosaccharides, general oligosaccharides, consist of same type of sugar moieties, forming kind of what is called as polysaccharide, the oligosaccharides attached to the membrane proteins and lipids can display extensive variability in composition and structure. So these oligosaccharides, they are not very long, they are 15 sugars per chain, and these sugars, they vary. They are not usually polysaccharide of one sugar moiety, but the sugar moieties can actually vary and can be attached to the protein as well as to the lipid molecule which is there. This attachment, the oligosaccharide attachment to the protein, is basically by two types of linkages. The two types of linkages are the N-acetyl glucosamine and N-acetyl galactosamine. So you can see here that you have a glucose moiety here. So this is the amino acid for a protein where it is attached. This is the polypeptide chain. So the glucose moieties is attached to the asparagine amino acid residue, and such a kind of branching pattern or an attachment pattern is called as the N-acetyl glucosamine. So you can have now multiple sugar units here, and you can have the entire polypeptide backbone here. But the asparagine residues is the residue where the glucose will attach and then will allow the rest of the sugar unit to be attached to this particular protein. In N-acetyl galactosamine, galactose is the moiety, sugar moiety, and then you can have various different types of sugar units attached here, and this galactose moiety is attached to the serine or threonine amino acid residue. So this is the polypeptide backbone chain. So at the serine and threonine residues, through the galactose, different sugar moieties will be attached. So you will find major these two types of glycosidic attachments, which is the N-acetyl glucosamine or N-acetyl galactosamine in the plasma membrane. These carbohydrate projections play an important role in mediating the interactions of a cell with its environment and sorting of membrane proteins to different cellular compartments. So again, when we will talk about the function of the plasma membrane, we will talk about the function of these carbohydrate moieties or oligosaccharide moieties, whose primary function is in these two processes. One is the interaction of a cell with its environment. They can interact with certain different molecules or certain kind of different things which are there, and sorting of the membrane proteins to different cellular components. So when we see that, when we talked about the endoplasmic reticulum, when we talked about the protein sorting, we saw that in the Golgi complex, certain oligosaccharide moieties are attached to the growing polypeptide chain, so that when they come into vesicles, they have specific receptors which are present, which categorically package them into certain vesicles, and these vesicles then target various membrane organelles. So the recognition of the protein molecules, that where to which organelle they need to be targeted, they need to be identified or sensed, and that sensing is through these oligosaccharide chains which are added as the proteins passes through the endoplasmic reticulum through the Golgi complex to the vesicular transport vesicles, which then target them to either throughout of the cell or to the lysosomes for degradation or to different organelles such as mitochondria, nucleus, etc. So oligosaccharide unit again plays a very, very important role, and they are attached to the plasma membranes which is there. One example which I can give right away of the presence of these oligosaccharide units and its role is in the ABO blood group system. So the presence of this ABO, the difference in this A antigen, B antigen, O antigen, what we talk about in immunology, is because of the presence of these different oligosaccharide units which are there. So A has a different oligosaccharide unit, B has a different oligosaccharide unit, and when both AB are present, it is said to be AB containing AB antigen, and O when neither of them are present. So there is a precursor molecule which is the H antigen, and which after processing contains different kind of oligosaccharide units. So then they are displayed onto the plasma membrane. They are oligosaccharides which are attached to the erythrocyte proteins, erythrocyte plasma membrane proteins, and displayed to the outside. And that is how they are basically, recognized by the different molecules. If we talk about the antibody, if we add the antibody against antigen A, this antigen A is an oligosaccharide unit which is displayed on the surface of the RBCs because it is attached to a protein molecule on the RBC. So it's a glycoprotein which is part of the erythrocyte plasma membrane which is there. And different antigens tend to give rise to the different blood types which is there. So this is an important part of what we talk about the carbohydrates which are present in the plasma membranes. The last composition, the last component is the membrane proteins. A membrane may contain hundreds of different proteins. Each membrane protein has a defined orientation relative to the cytoplasm which is there. These membrane proteins can be grouped into three distinct classes, distinguished by the intimacy of their relationship to the lipid bilayer. One are the integral proteins that penetrate the lipid bilayer. Integral proteins are the transmembrane proteins, that is, they pass entirely through the lipid bilayer and thus have domains that protrude from both the extracellular and cytoplasmic sides of the membrane. Some integral proteins have only one membrane spanning segment, whereas others are multi-spanning fragments. So these integral proteins actually embed into the lipid bilayer and they can actually pass through the bilayer, forming the transmembrane proteins which are there. And the two other kinds of proteins, one we have talked about is the peripheral proteins that are located entirely outside of the lipid bilayer on either the cytoplasmic or extracellular side, yet are associated with the surface of the membrane by non-covalent bonds. And third is the lipid anchored proteins, where the proteins are actually anchored to the lipid biomolecular layers. So you can see the integral membrane proteins usually containing of alpha helices, however there are exceptions. This is a peripheral membrane protein which are associated towards outside or towards the outer lipid membrane, and the lipid membrane proteins where this membrane proteins are anchored by such as a GPI anchored protein onto the lipid molecule by these certain moieties. So this is a protein which we are talking about, and this protein through this oligosaccharide moiety is attached to the lipid head which is there. So this GPI anchored protein is one kind of a association of the protein with the lipid molecule. So we have talked about the composition of the plasma membrane, and we have looked at the different kinds of lipids, proteins, as well as the carbohydrate moieties of the plasma membrane which is there. In the next, we will go to the functions of the plasma membrane that they perform in the cell.
With this note, thank you ma'am. Thank you so very much for giving us this session on plasma membrane. And dear friends, we are going to conduct one more session on the same topic, that is plasma membrane. If you have gathered maximum knowledge through this session, or if you have any queries or feedback regarding the lecture, then do write to us at info.cec@nic.in. The lecture is going to be uploaded on YouTube soon. So keep watching us, keep writing us. We will be meeting very soon. Till then, take care. Goodbye. Thank you ma'am. Thank you once again.